Hello everyone, I'm Suryan from Department of Microbiology. Today I'm going to discuss about Sds-Page. Introduction to Sds-Page. Polyacrylamide gels are formed by the reaction of acrylamide and bisacrylamide that results in highly cross-linked gel matrix. Sds-Page or sodium dioxyl sulfate polyacrylamide gel electrophoresis is a technique used for a separation of proteins based on their molecular weight. It is a technique widely used for forensic genetics, biotechnology, and molecular biology to separate the protein molecules based on their electrophoretic mobility. Principle of SDS-Page. SDS polyacrylamide gel electrophoresis involves the separation of protein based on their size. By heating the sample under denatured and reducing conditions, proteins become unfolded and coated with SDS detergent molecules, acquiring a high net negative charge that is proportional to the length of the polypeptide chain. When loaded onto a gel matrix and placed in an electric field, the negatively charged protein molecules migrate towards the positively charged electrode and are separated by a molecular sieving effect. After visualization by a protein specific staining technique, the size of a protein can be estimated by comparison of its migration distance with that of a standard of known molecular weight. It is also possible to blot the separate protein onto a positively charged membrane and to probe with protein specific antibodies in a procedure termed Western blotting. An important note: acrylamide is a potent neurotoxin and is absorbed through the skin. Take appropriate safety measures, particularly when weighing solid acrylamide and also when working with the solution gels. And use only high quality reagents and water for SDS-Page. Gel buffers and self-prepared acrylamide stock solution should be filtered, decased and stored at 4 degrees Celsius. Materials required: power supplies. It is used to convert the AC current to DC current. Gels. These are either prepared in the laboratory or precast gels are purchased from the market. Electrophoresis chambers. The chambers that can be fit the SGS-based gels should be used. Protein samples. The protein is diluted using SGS-Page sample buffer and boiled for 10 minutes. Running buffer. The protein samples loaded on the gel are run in SDS-Page running buffer. Staining and destaining buffer. The gel is stained with Kumasi stain solution. The gel is then destained with the destaining solution. Protein bands are then visible under naked eyes. Protein ladder. A reference protein ladder is used to determine the location of the protein of interest based on the molecular size. Protocol of SDS-Page. Gel preparation. Clean the glass plates and spaces of the gel casting unit with deionized water and ethanol. Assemble the plates with the spaces on a stable, even surface. Prepare resolving gel solution using depending on the percentage of gel required. Pour the gel solution in the plates assembled with spacers. To maintain an even and horizontal resolving gel surface, overlay the surface with water or isopropanol. Allow the gel to set for about 20 to 30 minutes at room temperature. Note: lemon must be the last ingredient added. Sample preparation. To a volume of protein samples, add equal volume of loading buffer. Boil the above mixture at 95 degrees Celsius for 5 minutes. Centrifuge at 16,000 into G for 5 minutes. The samples can be stored at minus 20 degrees Celsius or may be used to proceed with gel electrophoresis. Electrophoresis. The gel cassette is removed from the casting stand and placed in the electrode assembly. The electrode assembly is fixed in the clamp stand. One electrophoresis buffer is poured in the opening of casting frame to fill the wells of the gel. Prepare 30 ml of the denatured sample in the well. The tank is then covered with a lid and the unit is connected to a power supply. The sample is allowed to run at 30 mA for about 1 hour. The bands are then seen under UV light. Application of SDS-Page. It is used to measure the molecular weight of the molecules. It is used to estimate the size of the protein. Used in peptide mapping. It is used to compare the polypeptide composition of different structures. It is used to estimate the purity of the proteins. It is used in Western blotting and protein ubiquitination. It is used in HIV test to separate the HIV proteins, analyzing size and number of polypeptide subunits to analyze post-translation modification.